I'm John Spiro. I'm the Deputy Scientific Director at the Simons Foundation Autism Research Initiative. That's a program within a much larger foundation called the Simons Foundation, which is based in New York. And I've been there about 9 years. And our charge is to make a difference in the field of autism. We're learning a lot about genes that confer risk for autism. So the Simons Foundation has been a reasonably key player in this field in terms of helping to put together large cohorts, publicly available cohorts of people with autism, funding very large genetic studies to try to understand what's different in people with autism than in neurotypically developing kids. But what's really challenging is trying to understand, going from this list of genes, which we now have, so 5 to 10 years ago we really knew only a few genes that were involved in syndromic forms of autism, and now we have a large list of genes, but going from those genes to understanding behavior really requires a key intermediate step of understanding neural circuits. We should never forget that we ultimately want to understand a human condition, and we really do have to think about ways of studying humans in sort of very quantitative ways and linking that with the genetics. But I think a core area that really needs a lot more thought is the appropriate animal models, and there's a lot of discussion about using an animal model for an appropriate purpose. As we know, many of the animal models that are used for such good reasons, because it's so easy to manipulate genes and to measure behavior, like the rodents, are really far from perfect in terms of understanding sort of higher order brain issues. So we heard this morning from some work in transgenic marmosets and using monkeys, and I think that's an interesting path forward, but it's not without very, very steep challenges. I think there are so many exciting things that are starting to fall into place now. So one of them is an ability to interrogate the genome of individuals. So in the past, it was often sort of a group of individuals with a certain disorder and a control group, and now technologies are improving and the costs are dropping so that we can really ask very specific questions about individual genomes. So that's one part of it. Another part is just a vast increase in the sophistication that we can measure neural activity from awake behaving animals. When I was a postdoc 15 years ago, that just really wasn't possible. I mean, people were working on particular model organisms where you could record from head fixed and where you could get some interesting behavior, but the ability to actually get this neural activity in the context of natural behavior, I think, is going to be extraordinarily important.